all CIs.
Okay. Oh, good.
I don't know how many of them will go.
They show very nice. Toby has it over there.
Keep it up. Or have a cup of water. Hey, I'm doing good.
How are you? How are you doing, Carolina?
Good, how are you?
Kill any ducks?
Sorry, counts, guys.
I'll be back in window.
So you get.
You're not a. You're not Yeah. You have so good. Do you want to write like
Did you?
your name on it, like a little heart?
Very standard. Crazy. I think back. They tell you the. I think there's
Yeah.
Yeah, we haven't done that yet.
You know why? It doesn't have
So it's kind of like long way from the tracks. This was mostly just even if you're so it's just there's
I don't know what no, that's for our graduate students. What about you? Double extra credit?
so yeah. So it burns.
He is joining us today from Colorado State University, where he is the James C. Kennedy Endowed Chair in Wetland and Waterfowl Conservation, a position he recently accepted. Dave got his BS and MS from Untitled State University and his PhD from Auburn. He then did a postdoc at the Max Planck Institute in Germany, and then started at Utah State University as assistant professor there, and then, as I said, just recently joined the faculty at CSU. He studies all kinds of population demography questions in his lab, and while apparently his talk mostly focuses on SCOP today, broadly he'll be talking about how we can integrate different approaches and data to better understand population demography as well as management implications. His lab has done everything from songbirds to bison in different aspects of population demography, and he should give a great talk today. So please join me in welcoming him.
We'll see. So a few days ago, Sam emailed me and said, "Can I have a bio and a title?" I was like, "Oh no, I need to make a title before I get in the airplane." So I came up with this, and we'll see. You can let me know if it fits. Sort of a combination of catchy and content. The catchy part being the puzzles, integrated approaches to solving demographic puzzles in ecology. And I'll start by kind of starting the top of why do I think it's important to try and tackle demographic puzzles. And the importance could come from different angles. Some, given your field of study, some might be most interested in how individual variation in traits and phenotypes convey differences among individuals in fitness components, which when you boil it down are just vital rates. The processes of birth, reproductive output, growth, and survival from a wildlife or fisheries management angle are also fitness components. So an evolutionary biologist would be studying those same things. And individual differences in those give rise to individual differences in fitness that affects the composition of individuals and populations that over periods of time could lead to evolution. Or it could be you're interested in how environmental conditions and changes in those environmental conditions lead to changes in population level reproductive success and survival that affects the dynamical change in net abundance in a population that has long been the state variable of focus in fish and wildlife management that oftentimes leads to happiness or discontent of stakeholder outcomes, but all boil down to demography. Another level of ecology, you might be more so interested in species interactions and how those interactions between species affect vital rate performances of the interacting species that are the proximate drivers of species coexistence and exclusion that gives rise to biodiversity. That in turn affects nutrient fluxes and energy flows in ecosystems. So in my lab, I and students and postdocs, we generally focus on demography. And we use that as a lens to look into different realms of ecology and also conservation and management. But the central theme amongst our various projects is demography and how we can use it to address different, both basic and applied questions. Now, that's sort of just an introductory overview of what we do. There's a big question that's probably ubiquitous to many, many studies. You could change the word population with a different level of ecology, but there's a lot of our foci right now and funding is how are populations or communities or individuals affected by anthropogenic global change. And that's a really big question right now. And it's a very useful one to pursue, but it's also really big. And sometimes we come up with answers to it that are a little bit devoid of mechanisms and maybe sometimes even storytelling. I think it's important to take a slight step back and address intermediate questions, such are what are the demographic parameters that are most affecting population dynamics or community dynamics or fitness dynamics as global change is pushing on those systems, to get at one layer of the mechanisms so that we can have more rigorous answers to that really big question. So that's where, with some of my most recent research, we start with was this question: can we better study which demographic parameters are most affecting populations and population dynamics? To get at that, we kind of have to look at how can we tackle that question. There's different tools we can use to address this situation. Now, if we're talking about population dynamics, we're usually going to be thinking about population growth rate, and the most common one you'll see in textbooks, the one most commonly you'll learn about in a population biology course at a graduate level, is called lambda, and they put the subscript 1 to mean the asymptotic population growth rate. And what the asymptotic population growth rate is, a great kind of simple way to measure population growth. What it's actually applicable to are constant environments, these imaginary environments that do not change over time, and also that there's been enough time that the age distribution or the stage distribution or size distribution has enough time to settle into what we call a classic stable age distribution. So it's a great measure of population growth rate if those conditions are met. You might see where I'm going with this in that, well, oftentimes our world, especially with global change, is not like that. Now, a better tool set might be that maybe we'll try to tackle what's affecting the stochastic growth rate, just with the subscript S, that measures the tendency of a population to grow or decline in a world that's changing, that's variable, going up and down. Now, an often unknown caveat for measuring that type of population growth rate is that that noisy world has to be quote-unquote stationary. That means, yes, it's changing, but the mean is not, and the variance is not. That's what I'm trying to depict here in this graph. Sort of this bounded variability and some central mean. That allows the underlying structure of the population, like the age structure, for each component to settle on its own stationary distribution, and that makes it mathematically possible to measure a stochastic population growth rate. So this question of what's most affecting populations in a changing world from, say, Buich-Bottle rate, we could use that tool or that tool. Which tool would you rather use so far in global changes? That one? Or, don't be shy. It's okay, because maybe it's acknowledging a variable. I thought long and hard about that throughout my career, and I've done research and published stuff using both of these measures. But to be a scientist, I got to self-critique myself, and I come admitting that neither of these are appropriate. Because in a world with global change, I tricked you. We're experiencing non-stationary environments. Not only we, but the things we study. The fish and wildlife and various organisms, invertebrates and plants. No matter which aspect of global change it is, if it's land use, hydrology, or climate, there's trends going on. We're getting more or less of certain things, and in some aspects, like with temperature in certain parts of the world, they're getting more variable. So in this picture here, I'm showing what a world might look like over the past 30 years and maybe going into the future in some areas where there's some trend in the environmental condition. Maybe it's getting warmer or it's getting droutier or wet, and maybe the variability is changing. So that's a noisy world that is non-stationary. There's some trend in the mean and the variance, or both in this case. Now it turns out those two growth rates I just showed you do not measure appropriately population growth in this world. And for the reason there's not much known about how to measure population dynamics in this world, and thus not much how to accurately measure fitness or even community interactions, is that mathematicians don't like to deal with this. There's no long-term solution in a non-stationary environment, so you can't derive like a nice clean mathematical proof of what will happen. The best you can do is deal with some time frame of interest and measure what's going on. And we already have a measure of population growth. It's just this good old ratio of abundance at one time versus a previous time. You might just call that like an empirical population growth rate. For lack of a better term, I've called it a realized population growth. It's what populations are doing at each point in time, and you could measure a variance and a mean and a trend over some specified period of time. Now, is that important that we move into a different realm of measuring population growth and that might be in our textbooks or 99% of the literature? This is just a graph to show you, maybe it is. I've got here just a trajectory of abundance over some 25 years. These are the actual ratios at each point in time of that line, ratios of abundance, the realized growth rates. So at what percentage is the population going down or up each year? Down here, I have the asymptotic growth rates. So if I took the birth and death rates at each time step, put them in like a matrix model, and solve for the asymptotic growth rate, that's what I have here, and I have them at each year. What these ignore is the feedback between vital rates and population structure. They're just ignoring that. And that's why these are sometimes very different, like suggesting a population is pretty much stable when it's actually growing very quickly, or sometimes suggesting that a population is declining when it's actually increasing. So if you just kind of look at here, you'll see points that don't agree with one another. What that means is that the asymptotic growth rate can sometimes very grossly give you the wrong answer. And so if we're trying to understand what's driving changes in asymptotic growth rate, we might get an answer that's very different than if we look at what's actually going on. So that's just the first layer, meaning that how we measure population growth can be very different because they're applicable for different types of underlying environments. The asymptotic growth rate is really only useful for this kind of thing, but sometimes we use it and look at time series of those in a changing environment, even though it's not meant for that. So that case caused a dilemma. We had no real good piece of established research to tell us, okay, how do we go about figuring out at which birth rate at a given age or which death rate at a given age is affecting these things? We had no tool set. So we figured out, could we develop that tool set ourselves? And we eventually, through some hard work and finding collaborators that are much brighter than I, we developed that tool set. We call that, for lack of a better term, transient light table response experiments. The word light table response experiment is the tool for looking retrospectively and decomposing changes in population growth into its component vital rates. We use the word transient just to mean it works in non-asymptotic environments or these non-stationary environments. What these qualitatively, what they do is they help tell us which vital rates and which components of age structure, like the portion of the population that's old or young, how both of those contribute to population growth rates, and they let us do it for any type of environment. And without showing you the math, just trust me, we developed a tool that would help you figure out which birth rate or which death rate or growth rate most affects the variability in realized population growth rates over a specified time span. Some folks want to know that. What affects the variability of population growth rates? But for other questions, that might not be the thing you want to focus on. So we also developed a tool to figure out which vital rate most affects the sequential change in population growth across sequences of time. And that might be of interest when you really want to figure out what drove the population to all of a sudden start crashing, or what drove it to suddenly start rebounding or recovering. at key points in time. Now, you might want to ask something different, and the one I think that generally, at least for my interest, that I'm most interested in, is trying to figure out, okay, it's a noisy system, but I'd like to know about some trend and why the trend changed. You can measure a short to moderate term trend with a geometric mean growth. So I'll just use that subscript. And we figured out how you could measure which vital rates most affect a change in trend. In this example, from generally steady to crashing and then recovering. We could then go down and say, okay, at which survival parameter, which reproductive parameter at a given age was most affecting that. And then apply it to any real-world population that you would want. So kind of 3 flavors of breaking it down. You want to focus on the variability population growth rate? We can give you the answer for which vital rate's most affecting that. If you want to look at a change in the growth rate, we can give you a tool to figure out which vital rate most affected that, or changes in the trend. We can also give you tools to figure out which vital rate most affected. Does all that work that we did, I'm not showing you all the math behind it, doesn't matter. Well, we thought we need to show that. So we looked at a lot of life histories. So species with different life histories ranging from very low to very high juvenile survival, and from very low to very high adult survival. So down here you'd have really short-lived species, and up here you'd have really long-lived species. And we measured by simulating truth and then throwing our tools at it, we measured bias. And right around here in this sort of light yellow is no bias. And you might be saying, I'm just giving you a rigged figure here, right? How to lie with figures. Well, just make your scale really big and everything looks small. Now I make that scale big because we wanted to compare our new tools to the traditional tool that was being used in both the conservation and management literature. Regular life table response experiments that focus on asymptotic growth rates. So we simulated truth, used the old tool, and looked at bias. And we don't see really big bias across the board, but actually this is quite concerning. We see a life history pattern in it. We see high bias for certain types of life histories. Here in the southeast, these are the type of critters that lie into these realms where it might be biased. And then there are some species where your old tool, even though mathematically wrong, could still give you a fairly close answer to using our new tool. So is it which one's right or wrong? Well, in terms of the actual answer you get, it depends on what you're studying. And we're using approximate life histories. These could be even a little bit off. But we found is that you're never going to really get the wrong answer here. So might as well go through the effort of doing a little bit harder method stuff that'll give you the right answer if your world is like today and it's non-stationary. So that was all actually painfully hard stuff to get there. We think it might be an improvement, especially for certain species, but there's a caveat. These new tools, they not only require a nice time series of vital rates, birth and death rates across, say, various ages, they also require a time series of population structure. What do I mean by that? Like age structure or size structure, say if you're studying fisheries. Size structure if you're looking at plants. And I don't know about you, but in humans, it's not too hard to go out and measure the age and sex structure of a population. But in wildlife, when we go out and count things, it's hard enough to count things and account for detection, but it's even harder to figure out what age they are from, say, 0 to 30 or something. This is something that we know exists, but it's really hard to get direct data on. So then there's a conundrum. We're like, oh great, we have this new tool. We don't have this. Can't use the tool. So I'm going to transition now to thinking about how could we take another kind of new thing going on in demography that could help give us at least estimates of all this stuff we need to use to then figure out which vital rate is most affecting the system that I'm studying. And I'm going to get there kind of slowly. I'll first cover, what do we traditionally do? Well, if we're trying to get at population dynamics, one approach might be to go survey abundance. Count some stuff. When I tell people I'm a population biologist, they say, oh, you count stuff. Okay, I guess so. Sometimes we do that. We do count things. Those ratios of counts we give population growth rates. That's one way to do it. Another common way is we go out and estimate vital rates. Sometimes that's more expensive, so we might do that at a smaller spatial scale, like a study site. And we might do that with survival and some sort of measure of reproductive success, then combine them into some sort of population model. And then we'll try to project population growth rates from that. And if we're lucky, we might generally get a qualitative pattern that's somewhat similar, but most often, we don't get all the same details in these two different approaches. They should match, because we know birth and death affects abundance, and if we're counting abundance correctly, these two methods should give us exactly the same answers. But time and time again, when you talk to folks that have been doing this for 20 years, and they've done both of these, they say they never match up. And I recently had a colleague in Alaska say, every time I do a study like this, I get a population growth rate that's lower than when I count things. And then you have other people say, every time I do this, I get population growth rates that are more variable than when I'm estimating with abundance counts than when I'm doing vital rates. Theoretically, they should all match up, but in reality, they often don't. And you get wars that say, this is right, that's wrong, or this is right, and that's wrong. And really, it's just a he said, she said argument. Reality is probably there's bias in everything that we do. And we need to acknowledge that. It could be, and I wanted to include this because maybe that's not the way you do things. Maybe you do things commonly in a fisheries world, especially marine fisheries, more of a reconstruction method. We have lots of dead things, like lots of dead fish or dead animals, and we can age them. If we have a distribution of age, we can back calculate vital rates. But here again, we get answers that sometimes differ. This is just a long way of saying, everything I've done in my career, I always feel like I'm trying to take a hammer to the puzzle and shove things into boxes where they don't quite fit. And I don't know why they don't fit. But then along came a new approach to doing population analysis, and they're called integrated analyses. Or more specifically, integrated population models. And here's the key difference. Instead of saying, we're going to do things one way or the other, just look at count data or just look at vital rate data, it's a way to acknowledge that all of these are informing a common process. Remember the good old BIDE equation. Abundance changes from one year to the next as a function of at most 4 demographic parameters, or immigration, death, and emigration. It could be that then they're structured and we need to use like a fancy matrix model to accommodate age and size structure, but they're all balance equations. It could be you can ignore migration, but it's still a balance equation. One thing, simply said, abundance and vital rates leads to change in abundance. So what integrated analyses do is they say, take all that data and analyze it simultaneously by acknowledging which pieces of this equation they form. Now, what that does is it helps reconcile bias that exists in these data sets in a way that balances it out. So it might upweight bias on one side and downweight bias a little, and basically what it does is in the he said, she said argument, what's better, it comes to a consensus. Doesn't mean it gets rid of bias, it just means you come to a consensus. It also does a few other things. It gives you the ability to do things you wouldn't be able to do otherwise, which is, let's say I have some vital rate, like for survival, but I'm missing reproduction data. You can still proceed, as long as you've met other assumptions with the analysis, because what that is like doing is solving for one unknown when you have data about the left and the right side of the equation. When I have counts as sequences of time, and I have some of the viral rates, but I'm missing data for just one of them, this is just sort of a hokey example here, I can estimate that other viral rate. Or, one way to say this is, I don't have any data for reproduction. But if I got data for everything else, I can still get estimates of reproduction. You could use that in planning purposes to save money. Maybe budgets just got cut. And I'd still like to get an answer to this. This would allow you a framework to figure out, well, if I design things appropriately, I can still get the answers I need without spending any money on that extra thing. Here's the one for me personally that connects the work to my previous slides is the integrated population models will give you simultaneous estimates of a total abundance, how big is my population? Population growth rates, is the population going up or down? Latent population structure. Latent is just a fancy stat term meaning it's there but we don't see it. It'll give me estimates of the age or size structure of the population. That and the vital rates. I get the whole package from that. That then will let me, because I get estimates, that it'll let me use those tools I just showed you, those transient life table response experiments. I'm going to get a time series of estimates of vital rates and population structure. I have all the pieces of that puzzle that I need to now apply those transient life table response experiments. I just put a couple of review papers up there for sort of the wildlife folks and the fisheries folks that talk about the history of these integrated population modeling approaches. I got into it when folks like Mikkel Schaub started publishing a lot, and then I realized the marine fisheries folks have been doing this stuff since the early 80s. It's been around. Of putting various pieces of data that should agree with each other together in one fluid analysis. Now I'm going to segue into a very specific example of where we take these two relatively new tools, transient life table response experiments and integrated population models, and we fuse them into one fluid analysis to try and come up with an answer for why in the heck did my population go down or up. And I'm going to use a real world example that basically is where I got started. It's what Beth did her PhD on, Lesser Scott. The reason anyone puts any dollar to Lesser Scott is because of this. These are the dynamics of total duck abundance on our continent, approximately in the core areas where they breed, over the past more than 50 years. They go up and down. Their ducks, when there's droughts, they go down. That's not surprising. And when it's wet, they go up. That's what ducks should do. And that's what they've done. And right now, we have really healthy total duck numbers. Scott have not followed suit. They kind of, in the early days, were doing what other ducks went. When it was wet, they were fine. When it was dry, they were not. And then they rebounded. And then something around the early 80s switched. They started declining. When other ducks rose, they kept declining. In recent years, we've seen some rebounding, but they're still below the long-term average. And as they've declined, that long-term average keeps going down. So they're one of just a couple of species of the primary survey ducks that are not doing so well. And for a decade, we had folks who were, as arguments, it's toxins, it's this, it's that, and we'd have little little studies that would help give us some evidence of what might be going on. But we still didn't know if it was primarily a survival problem or a reproduction problem, because we did not have that kind of data at large spatial scales. So we thought maybe this integrated approach could help us gain some traction on that. So what we did is we used kind of monitoring data at large agency scales. And I'll show you in just a bit what I mean by what those data sets are. These are data sets that give us indirect information about vital rates, but maybe not the very specific thing we're after. We used a Bayesian approach, so we would use prior distributions, and we decided to be fully Bayesian and actually use prior information to create our prior distributions. What we did is we took all the various student-led projects that looked at vital rates and averaged those to create prior distributions for survival and reproduction and how reproduction varied across age classes. In a Bayesian approach, we combine those priors with our big data that's at the scale we're interested in. I should have said we're interested in continental scale dynamics to try and get some sort of consensus. And then we combine that into a structured model with two age classes, sort of first-year breeders and older females, and the same for males. And our goal was to estimate vital rates over 50 or so years, population structure, population growth rates, and how to figure out which vital rate was most affecting population growth rates and any trends. So where do we get the data? Here I'm using F for fecundity or reproduction, S for survival, N for abundance, T for time, and X for age class, young or old, and sex category. Well, there's always been some banding data, but it's been, the waterfowl people always called it weak because relative to mallards, there's a lot less got banded, and they're not banded contiguously across the continent, but there is some banding data. We thought, well, on its own, there are some issues with it. It's getting better, but we thought together maybe it'd be useful or helpful to use that banding data to inform survival. But then there's also this really cool piece of data that on its own has some biases. It's called the Parts Collection Survey, or the WINGB, where citizen scientists that have been doing this for more than 40 years, a subsample of hunters are requested to and participate in the survey, where they send in the wings of all the birds they harvest. Then every year in each flyway, state, federal biologists, and every now and then a few volunteers get together and they categorize these wings down to species level, but more importantly here, they categorize it down to sex and age category. It gives us like a sex and age ratio, and we know there's bias in that because young birds are more susceptible to be shot than old birds. And sometimes certain sexes are more susceptible to be shot. But when we combine that with the banding data also gives us these relative estimates of relative chance of being shot, recovery probabilities. We can use those to adjust the data here and get at a less biased age and sex ratio. Is now giving us indirect information about, through the age ratio, the reproductive success, and through the sex ratio, relative survival across the sexes. So on their own, each piece of data is not the best we like, but together they're giving us more information. Then when we combine that in one fluid analysis with the count further helps reconcile the differences amongst the data because we know the balance of birth and death should be affecting how much abundance goes up and down. And we get the total count, not count by age and sex class, but just a total count by these aerial surveys that have been conducted across this massive spatial scale since 1955. Where does the information come about age structure? It comes from getting age structure in the vital rates. That then indirectly helps us break down abundance and estimate it according to age and sex class. So these are just boxes showing these are how we use the data. We use it all together, and wherever boxes overlap, information is being shared across the data sets. So when we analyze them together, the data are sharing information to help inform this common process: birth and death affects change in abundance. These are just a snapshot of the kinds of things we get out of it. These are survival probabilities for males and females, adults and juveniles, across since 1957 up to about 2013 was our last year we had data. Remember, the population starting about the early 80s started declining quite precipitously. One thing just to note here, we never observed survival of these adults to be going down. And if anything, actually, where I have a blue line means a statistical change suddenly up or down, or a slope that's significantly going up or down. Males generally were pretty stable. In the recent years, they went down a little. Don't care too much because there's a lot more males than females. They're not limiting the population. Female survival has been trickling slowly upward while the population was going down. I probably wouldn't even have to do some fancy analysis like I'm going to show you results from. Right now, I'd probably say, I'll bet female survival did not cause that population to go down, because it never went down. But I'll go further anyway. Juvenile survival was kind of interesting. It was trickling up. At about that time that population started to crash, juvenile survival tanked, but then it recovered. So I don't ever see a pattern in these things that directly mimics the pattern in abundance. But remember, abundance is a compromise amongst survival across various categories and reproduction. So if I wanted to look at what most drove this variability in the population growth and not really pay attention to ups and downs, what we found is that the variability in juvenile female survival was most effective then. Even though models show that adult female survival, all else equivalent, had the greatest potential to affect the population. So the vital rate with the greatest potential to affect the population was not the vital rate most affecting the actual population. And we determined that using those fancy transient flight table response experiments. Now what I also want to show you is that integrated population model also helped us estimate reproductive success. This is just reproductive success or reproductive output of the older experienced females, same pattern in the young females. It was going down for many years. Ever since the early 50s, it was going down quite sharply. and has never recovered. It's starting to even slowly go down even more, but not as abruptly. So it was going down before the population abundance was going down. And what's probably going on here is one vital is going down, others were going up, and they were compensating for each other for quite a while until another vital rate dropped. So you had a double whammy that then led to the population from kind of holding stable but wiggling to then crashing. What we found is that it was that long decline in fecundity that most affected this, but it was also a close second, the juvenile female survival crash, that couple that led to that. And because fecundity has not recovered, and it has taken juvenile female survival a while to recover, that we do not see, we have not until recently seen in recovery and abundance, and the recovery's been kind of slow. And the new data we just got showed, well, it's not going up, it's just sort of hanging low right about down here. So reproductive success combined with juvenile survival, those are what dictate recruitment. So what this whole exercise helped us learn is that hunting of these adults is an unlikely culprit of the decline, but it is the thing that management has targeted. There's been severe restrictions on how many scopic have been harvested. Why? Because we can do it. We can change regulations, but this shows us it hasn't really done anything. What we would suggest from our results is that focusing more research on what's been affecting recruitment, the combination of producing offspring and getting those offspring into the breeding population, would provide more information about what has affected the scaup population and what we could do to recover it. So that's an example. The world's been changing. We use this to break it down and figure out which part of the life cycle was affecting it. And then the next step would then be to go further, we haven't done that yet, of figuring out what is it that's most affecting recruitment. That'd be a next layer, using retrospective data and new studies to do that. Now, that's one way of doing an integrated analysis, focusing on population index. But you might be studying some system and you say, I don't care about populations, I'm interested in something else. It turns out integrated data analysis has been around for a while. Statisticians have used it, but it's not super common. You rarely see it in a stats textbook, but it's very general. You could use it to address any question where you're doing a statistical analysis. And the generality of sort of is like this. Let's say you've got some study area, and everywhere you have data D, but everywhere you think it's inaccurate. For some reason, it's just you're not collecting the data perfectly. Maybe you don't have enough money to collect really good data across a large area. But in a few areas, you're able to spend more money and also collect a more accurate source of data about the thing you're trying to study. You fill in the blank of what the thing is that you're trying to study. What an integrated data analysis is generally doing is allows you to use more than one source of data in one fluid analysis. I could use both my inaccurate data and my accurate data together. Here's one example of how you might use it. It's not the only way you could use it. Is you could say, well, I'm trying to understand some process. I don't really have great information about it everywhere. I have some data, the data is sort of inaccurate, but at some layer I'm going to have to account for that error in my data. If I use and replace the unknown process with known, really accurate data in some locations, I'm now getting a direct map about what the data should look like and what they actually look like. I'm basically using it to get a correction factor in one fluid analysis. So I'm not taking one data and correcting results. I'm doing that correction factor in one fluid analysis using my hierarchical model. You could do that for any kind of response you wanted. You could do it in social sciences, economics, you name it. This was recently done here right out east. Guthrie Zimmerman and colleagues had a scenario like this. They wanted to be able to estimate wood duck abundance. Wood ducks live in areas where you can't be flying airplanes and counting them because they're in these cryptic locations, or habitats that are thick that make them hard to detect. So you don't really have these aerial surveys where you count things from an airplane really nicely like you do in the prairies. You probably don't want to be flying planes at low altitudes over rugged treed landscapes for obvious reasons at low speeds. So there is the good old breeding bird survey. Citizen science survey, people go out and drive a route, get out of the car, and look for things and listen for things and record them. These are the different bird conservation reasons here. You have that data. It's probably that kind of data. We worry about breeding bird survey data. It's inaccurate in various ways. There's observers that change. It's not like the most high quality data. But eventually, this Atlantic flyway breeding waterfowl survey, not from the air but from the ground, was developed to try and get more rigorous or more accurate data for certain species. But it's smaller in spatial scale. It only exists in these hatched areas, and it only exists at certain locations within there. But it's this scenario where basically in some areas, they had a broader scale source of inaccurate data and a smaller scale source of more accurate data. And they did just this. They fused them together in an integrated analysis to be able to estimate more accurately the wood duck abundance not only there, but across the whole area where you have breeding bird survey data. So using, in one analysis, sort of the correction information you're getting here, and then extrapolating that throughout the east. That then allowed them to look at population dynamics. Now they got abundance of wood ducks across space and across time, and go further. Now wood ducks, some wood ducks, not all, are like these other critters that I think occupy your wetlands and coastal offshore zones at some point in the year. So they're migratory. Some of these undergo long distance migrations. This is a new topic I've gotten quite interested in. Because I study birds, but for many times we've looked at breeding population dynamics. That's how our surveys are set up. But migratory animals don't spend their entire life cycle sitting in one spot like my colleagues do that study plant population dynamics. I envy them. They don't have to deal with all these complications. Migratory animals move habitats across the year, and they do this every year. And as they're doing this, they're shifting in a way they're going to other planets. Different types of habitats, they're very different than where they were the previous season. They then have to, I'll skip over that. They gotta go south and north, and they need to find new habitats as they're stopping over during migration, and then choosing a wintering area, and maybe even within a season, they're moving around a lot. They're undergoing aspects of habitat selection, phenologies of departure and arrival across very long distances in very different parts of the world. At key points in this life cycle, maybe all of them, but definitely all the aspects that involve migratory and stopover aspects, they're having to make complicated trade-offs about how much time to spend foraging and getting energy, when to think about, man, if I eat all the time, that damn hawk's going to kill me, when to think about self-maintenance and getting away from individuals so they're not getting a disease, not getting shot, not getting preyed upon. Various aspects relating to being vigilant. And then they need to think about, when is it time to just get the heck out of Dodge and migrate and start spending lots of energy? So they're undergoing these various life history trade-offs. The strengths of these trade-offs are fluctuating across the year. But that's going to affect the condition and health. Not only the fat condition, but other aspects of physiological condition that can affect seasonal survival, and this can feed back to affect reproductive success. One thing that connects migratory species is they're carrying out this annual life cycle at different locations, and what this can give rise to are these demographic and physiological carryover effects across the life cycle, whereby something that happens to this animal in the breeding season can now affect their condition and survival in the winter. Or more commonly, we look at how wintering conditions affect the health of a bird or migratory animals and how it affects their reproductive success. But these things occur across the entire life cycle. We always have known, we always think we've known that occurs, but I think it's important to start picking this apart in more detail and trying to drill into the mechanisms of how these things operate and how they affect population dynamics. The reason I think that's important is that those answers, maybe they're overly nitty-gritty things that some might think, can feed back to management and help us think about, do we want to allocate our dollars more to breeding habitat? Is that more limiting? Stopover or wintering habitat? But also part of this equation, if it's a species that has some offtake, whether that be harvest or bycatch, how much do we invest in that? Because it's all taking place in that annual life cycle as things are migrating. So there's both basic and applied reasons to think hard about that. Another reason why we don't know lots about the detailed mechanisms is it's hard to follow organisms across long distance migratory routes and know what their condition is and what their demography is. Now this comes back because I think this is where integration of data is going to, in the future, help us drill into these mechanisms. We can combine really gross scale inaccurate data with very fine scale accurate data, but with maybe various sample sizes. And just brainstorming a bit, what are the kinds of data we might want to use to study things like carryover effects across long distance migrations? We might want to use some stable isotope analyses just to figure out where origins of animals are, where was their breeding area, and where are we finding them in the winter. There's some information in that about where things were and where they are across long distance migrations. If there's lots of animals been marked like birds, good old banding data has long told us a lot about origin data like that. That's how the flyways were figured out. But there's a lot of things that happen in between where an animal's banded and where it's recovered that we don't know about with that data. At a finer scale, sometimes we mark animals and we can re-site them. So mark, re-site across geographic areas, giving us information about movements and also demography. But generally our sample sizes are not too huge. There's also like expert opinion surveys. This is an example of the mallard migration, where biologists sort of say, here's where the mallards are. That's giving us information, sort of like eBird, about the flow of relative abundance. Now, more detail with technology, this is something I've become interested in, but I'm just envious of everyone here out east, is the MOTUS system, M-O-T-U-S. It's sort of like combining new and old technology. You put a VHF transmitter on an animal, but they're small because you're also combining this with permanent station receivers that are receiving these signals as a bird migrates and flies by. It doesn't tell you where the bird is, but it tells you kind of plus or minus 10 kilometers where it is. And for most migratory questions, that's a lot more information than we might have had in the past. This is useful at least because these stations are located everywhere from Canada down to Mexico. If you go west where I live, there's one of these. I don't get it. There's a lot of dollars in California, and there's not a single MOTUS station in California, not even at Point Reyes Bird Observatory. There's one in British Columbia. I think those offer enormous opportunity to look at how animals are moving, when they're moving, and also what's their survival between these stations. Now, it doesn't tell us exactly where they are. GPS transmitters have been the way to go for telling us where animals are. They've generally been pretty costly, and for things I study, I get worried because they're like 1/4 the size of the bird, and now we're studying the movement and demography of handicapped birds. And I don't like that, for both ethical and scientific reasons. These are called pinpoint GPS transmitters. That's a US dime. These are GPS transmitters. They're getting smaller and smaller. I think I'll open up the ability to follow larger, and they're also getting cheaper. Larger samples of individuals, where they are exactly on any given day, maybe multiple times a day, and also how they're surviving. Now, ultimately, we'll have technologies that can even read, and you can do this for really large mammals, their body, and how that's changing. But this is just sort of one example, brainstorming of how we can integrate sources of data to get at these challenging questions. And for this topic, ultimately tried to solve that puzzle of carryover effects and demography across long-distance migration, and what's driving it, and why is it relevant for management. So I'll close with that. I just want to thank you all for coming. I could not leave without thanking this individual, James C. Kennedy. He's made the position I now hold, the position that Dr. Kaminsky holds. I thank him a lot because he's been instrumental in sharing information with Jim and establishing now 4 endowed chairs in waterfowl, which a few years ago had never been part of anyone's dream. That's why I'm here today, and get to talk to you and so on. Maybe go drink some beer. I can take any questions. Okay.
Excellent presentation, as always. Returning to the scope issues.
Sure.
You know, you don't have to go
to show a decline.
So, I've been around through most of that period of
Yeah.
ups and downs and declines. And you showed us some demographic dynamics,
Yeah.
if you will.
Yeah.
Age, sex, survival, undertake. But where's it happening?
Yeah. We don't know.
The question I want to know is, what is the agriculturally impacted prairies, parkland producing now in terms of scaup?
Yeah.
Similar to pintail. Is that the bottleneck?
I don't know. That's kind of where I got later with this migratory stuff. If we want this at this level, we could figure out basically birth death stuff. But they're migratory. And it doesn't mean that because it's reproduction, it doesn't mean that it's something on the breeding grounds or the core of the breeding grounds that's driving that. A lot of them breed in the prairies, a lot of them breed in the boreal forest. They're right there, there's a complicating issue. They all pass through the prairies and are getting energy. So it could be, that they're not getting enough food to make eggs or make them on time. Maybe they're delaying reproduction, now phenology is delayed, and now their ducklings are not overlapping with their preferred prey. That's one hypothesis. In terms of the more, getting towards the more ultimate mechanisms, we're still unknown. I think what this does help is it helps sort of eliminate a few mechanisms and helps us focus on still a fairly large suite. It's an advancement a little bit. It's not all the way there. I don't know if historically we are still going to have enough data to drill into what the details were. If I wanted to do that in one system in the duck world, I'd probably do it with black ducks. And I would do that because they have all the same data, but they also have strong wintering banding data and strong breeding ground banding data. So you have, if you band at 2 seasons, you can get at seasonal survival. And so now you get at some of the things of where is it happening? And then I'd throw about a lot of these nice transmitters on those things.
I can't recall seeing if anyone's partitioned out the BPOP estimates for scaup from the prairies versus the boreal forest versus Alaska.
This one.
Okay.
Yeah.
Tell us a little bit about that.
So
What's happening in the prairies?
scaup are actually increasing in the prairie parkland
It's what?
region. Scaup are increasing in the prairie parkland region based on the BPOP surveys, and they're decreasing in the boreal forests.
Really?
Something I didn't get into is there's enormous potential, and actually Richard Chandler at UGA is taking these and making them into spatial integrated population models. So drilling into the stuff we'd really like to know is, are they going up and down and where? At fairly detailed spatial levels. And that's using things by combining resources like telemetry data, sort of, or location-specific counts with location-specific banding data. There are ways to then extend this into spatial questions and actually sort of get landscape-level population growth rates, like heat maps, not where things occur, but how they're growing, their growth rates across landscapes. That's for the future, I think. But you're exactly right, Rick, because we still don't know about where it's happening and why it's happening. Another thing to note, it could be, and it actually, I didn't talk much about this, different things drove this than drove that. So it's sort of a reminder that focusing on only the thing that caused a historical decline might not be the thing to focus on in the future. Demography is more complex than counting stuff. So I have horrible elevator talks about what I do.
It's a great presentation, and I have one question about your integrated analysis. As you mentioned, that can be useful in many methods and fields. So we basically use, as I understand, some points that we have precise sampling to calibrate other points that we believe are not as accurate as those points.
Yeah.
So that part is great. But I was wondering how we can validate those.
Yeah.
Like how actually you're not forcing them to like replicates.
One just has to be careful of these because things can be going on under the hood, as we say, without you knowing it. So one way to validate is to do, without actually doing expensive studies, is to do simulations to look at what if I have this error in this data set and I combine it now? Am I getting an improvement or just making things worse? And you can show examples where you're actually, by combining things, you're making everything worse. So you gotta be careful when you do this. You gotta think logically, biologically, and also about what you might be doing inadvertently. Other way to do it is to do detailed, focused, specific studies. You know, maybe not huge scales is your inference here, but testing things. Do almost sort of pseudo experiments in a way to validate things like that. There will never be a replacement for nice studies like that. And I don't think these should replace anything. They should just be another tool in the box to use, to consider in some cases, but to consider it carefully.
Yeah, because they can be really useful. You can't always sample everywhere. Yeah, great, thank you.
They can also be very dangerous.
You mentioned it a little bit. To what extent can you incorporate eBird data into these models?
I've been thinking about that very closely because there is a great source of spatial count data, but I think it's one that has to be looked at closely with scrutiny about validation. I think there's a few types of data sources where one can, again, integrate data and just simply see, if I take two data sources on their own and I get answers, say population growth rates or relative movements of abundance north and south, and I look at the answers I get from those two data sets, and then I say, what if I throw eBird in? What does it do to this result when I think that data is accurate, like the higher quality data? Is it improving my precision and generally just giving more spatial detail, or is it really changing things like growth rates? Then I might worry about bias because it is, you know, citizen science without, you know, it's not like quite like breeding bird survey where there's a protocol, a really strict protocol. It's a little more, hey, just tell us everything you've got, and people can lie and all those things. But I think there's data sources out there to, in a way, do these validation things. Like in the waterfall world, we have these aerial surveys. We could combine eBird data to start figuring out where are the consistencies and where there are some wild differences. To then be able to think, oh, is eBird going to be useful to give me more fine-scale spatial things, or is it just going to throw everything off? So I think that's generally, qualitatively, how we can think about bringing your eBird data into it. But I think we have to be, there's got to be a lot of studies that look at the scrutiny and the validation of it. And my guess is, talking to a close colleague out in Oregon, it's going to vary by species and life history, unfortunately. It's not going to be ubiquitously applicable across all species. Ecology is noisy and complex.
Is there data that covers, or do you have the data to look at the migratory patterns and as they change, or as they might be changing specifically due to climate change or whatever, in terms of the extents north and south?
You know, not perfect, but there's some information about that, at least in the ductile banding data. We get kind of longitudinal information quite well. Now, where they're, you know, they can be, an animal might be trying to go here, but they get shot here. But if you look at kind of past the individual level and it's sort of aggregations, there is information there, as long as you look at everything at a relative and not absolute scale. Good old banding data gives us a lot of information. The key is getting recoveries. You can't just band them and throw them out there and never get a recovery. You need recoveries.
All right, well, I think everyone's heading down to Nick's for a beer. I encourage you to ask Dave about what happens when you combine beer and fires in an Arctic field camp. Excellent story to open up a conversation with. Yeah, let's give Dave another round of applause for Thanks, Ellie.